So this is a consolidated web page for what the research topics and what the research fellowship program is all about. So when we go down, I think you would have gotten a lot of introduction about how different students and researchers can make use of big data while performing different analysis. And what do I mean by that? Let's go down. You will get these specific advantages of when you sign up for the research fellowship program. So you have curriculum that's specifically designed for your research area and for your research interests, and you have your own self-paced learning, and most of the courses and most of the resources that we are going to share with you in the research fellowship program is asynchronous, meaning you will learn at your pace, you will do this at your pace, and then we will be there to discuss with you about your progress and about how to proceed further in the research fellowship program. And you will get hands-on with big data as expertly demonstrated by 3 students research topics. So all of them obtain data from publicly available forum, and all of these data are big data, big omics data, and they used this big omics data to address different parts of the biological queries and questions. And you will have access to expert mentors like myself, like Dr. Mohit and others. So when you start learning, you will get immediate feedback from us, and we are accessible to you offline. And also we will meet you once or twice during the week, during the scheduled research fellowship meeting. And also if you request for one-on-one meeting for some specific cases, then we will offer that also. So when you go down, what are the different areas in which the omics data and multi-omics data in general are being used to address specific research questions? You have a variety of topics that uses omics data as of now, which will include genomics data, transcriptomics data, epigenomics, metagenomics, and these are only few that we are targeting or that we are actually focusing on. You have furthermore, you'll have proteomics, you have metabolomics, and etc. A lot of other omics data types are coming up in trying to address different areas of biological research. So, and we have actually this site, this web link gives you some of the major applications of omics data analysis to different areas of analysis that is described, that is focused. So some of the obvious focuses on infectious diseases, and the pandemic era that we are currently living is a perfect example for what omics data can achieve in addressing infectious diseases case. And we all know different terminologies related to SARS, like variants of concern, variants of interest, how does it spread, like what are the different lineages, right? And what are the mutations that actually characterize variants of concern, these variants of interest. And all of these analyses are direct results from genomics data and transcriptomics data analysis. So when you use the whole genome sequence of the virus and try to compare it with the other strains and other variants of this same virus, you will get a lot of information. And this is just one example of what you can achieve from sequencing the whole genome sequence of the virus. And similarly, this can be extended to transcriptomic landscape analysis, which will characterize how a patient is going to respond to the onset of the disease or to some drugs that is going to fight against the progression of this disease. And these are just 2 omics data types that is trying to provide us with a lot of information in addressing some of the key questions in infectious diseases. And we can again do this in metagenomics data analysis and epigenomics data analysis, which are also extremely important omics data type that are coming up. And I would like to stress the importance of metagenomics data analysis, where the gut microbiome or the microbiome estimation of the human gut is being linked to various other disease onsets, like cancer, like allergy, like even neurological disorders and pathological states. So that level of association is being exploration of that level of association is being carried out at various different labs, and this is one of the interesting and upcoming omics data types analysis methods and questions that we can answer through analyzing and observing patterns within the metagenomics data. So when you go down, we have a lot of project examples on Omics Learn, which focuses on different omics data analysis in trying to address key questions in infectious diseases like SARS, like malarial infection, like the SF9 cells will actually tell you about how to identify viral contamination in your samples. And Ebola virus epidemic is an excellent example of how genomics data was used to track the progression, disease progression and spread of Ebola virus. So when you come down, the application of omics data analysis is at its peak to oncology because of the requirement and because of how omics data characterizes tumor, cancer, and generally the disease progression and its effect on various types of patients and various types of various types of races and ethnicity. So what we get, part of the reason why it is required, because of the existing traditional methods of identifying, characterizing, understanding the disease progression of tumor and treating the disease, tumor disease and cancer as such, is not sufficiently effective. So hence we had to resort to transcriptomics or genomics data analysis, epigenomics data analysis, and etc. So when we have a wealthy of such information and big data, it is natural that methods that are specifically designed to analyze patterns from these large quantities of data, like advanced statistical methods involving artificial intelligence, deep learning, machine learning, will come in handy. So they are also at its most useful when you are trying to use a large volume of data like transcriptomics data from lot of patients as compiled in a wonderful public repository like the Cancer Genome Atlas or TCGA. So we also have a NCBI database where the SRA data will provide you a lot of genomics information and genomics data where you can focus on, for example, genome-wide association studies, targeted loci studies to understand the specific genes and driver genes that are that could be used to answer and that could be used to explore the onset, disease progression, and the treatment response of different tumor from different cases. And there are also efforts in understanding cancer from integrating multiple or multi-omics as such, from maximizing the information and also integrating the omics data type with a wealthy of cancer history, patient history database and patient history data and the treatment information data and genetic data, genetic history data generally that most of the translational approaches will acquire at the time of diagnosis. And we can also use traditional methods like using structural biology and omics data integrated into structural biology for developing different drug targets and drug molecules like chemoinformatics data to approach the translational problems of finding and identifying targets for these cancer research, and also finding and developing treatment strategies for targeting these specific targets that we identify from structural biology efforts. As I mentioned in the previous case, metagenomics is one of the upcoming area that has been linked to majority of the onset and progression of disease from various other parts of our body. And this doesn't stop at cancer, and this doesn't stop at gut or chronic diseases development. So again, to give you a few examples of what are the different projects that we have in Omics Learn, we have a project that focuses mainly on the immune response of the cancer to different immune therapies, and we have projects that deals with mass or huge volume of data from the Cancer Genome Atlas to identify risk factors that are associated with the liver cancer. And we have a project that actually tries to relate omics data with the treatment response data that is observed from grafting the tumor into model environment like mouse model and like cell line models, etc. And that will straight away form, and that will straight away lead us from understanding oncology through omics data analysis into translational aspects of oncology in precision medicine through the application of omics data analysis in treating cancer. So for this specific aspect, or for this specific research topic, then we have one project, or we have multiple other examples with this specific project, modeling cancer precision medicine, which I was talking about, where the cancer cells were grown in a cell line in where they are grown in cell line environment, and they were treated with multitude of different treatments, and their response to different treatments through increase or decrease in the volume of the tumors are observed. And this treatment response, is directly linked with multiple omics that characterizes these tumor cells. And by linking these omics data type and a treatment response, we can identify which treatments are effective and how we can use omics data types to effectively or to accurately identify the effectiveness of this treatment. And this will fall straight into patient stratification in terms of which patients, or what are the subset of patients that will respond to a specific treatment, and treatment stratification in terms of what are the possible treatment options that I can suggest to a patient who is likely to respond to several different treatment options. And this doesn't stop at treating hosts from human, or from majority focus on Homo sapiens and human. This also extends to agriculture, where I believe the analysis has a lot to offer, and the application has a lot to offer in terms of understanding, for example, drought resistance, understanding, for example, salinity resistance, understanding, for example, heat resistance of crops, which naturally have acquired these characteristics since they are being exposed to these harsh conditions. One can characterize these plants using, again, these molecular characterization methods from omics data types analysis, and then try to apply whether bringing about such changes in a different plant can actually result in developing resistance for drought, developing salinity in resistance, and also whether can we transfer the ability of plants to survive in harsh condition by changing the omics landscape of these plants. So such efforts are actually actively going on in agricultural area. And again, to stress on the application of metagenomics data analysis to a wide variety of biology, and in general, people usually take the soil samples, understand the microbiome population of the soil samples from different parts, right? From, for example, from a region which receives very low rainfall, or from a region which receives very high rainfall. So we can understand what are the different bacteria, fungi, and virus that makes use of these conditions and effectively to help the plants grow at its maximum potential. So these are different areas in agriculture which are actively upcoming and developing currently, and both in basic mechanistic research and also in application oriented research in agriculture, in plant biology generally. And neuroscience is also one of the field where people have tried to understand, or people have tried to apply omics data analysis from samples from brain. And there is a practical difficulty in acquiring samples from brain for live patients, and hence now there is a lot of effort in trying to relate to neuroscience and neurological disorder from samples from blood, or from samples from gut microbiome, etc. So and advanced analysis methods and data science methods are helping us making these connections that are usually not observable using traditional analysis methods, and evaluating them for the statistical verification or statistical significance of these connections. And transcriptomics data analysis, as I mentioned, is one of the most used omics data analysis, or one of the most applied omics data analysis to wide variety of different questions related to human translational biology. And neuroscience is not agnostic to that also. So traditionally, people extract samples once the patients have passed away from the brain cells and compare it with the healthy patients who are age matched, gender matched, ethnicity matched, so that we are eliminating a lot of other confounding factors in trying to understand the gene expression landscape of neurological disorders compared to healthy patients. So when we extend such analysis to metabolic disorders like allergic reaction to gluten, or like allergic reaction to some specific metabolome, or some people will have acid reflux. So in a lot of these disorders, and some people will have difficulty in filtering salts in Sydney. So in a lot of these disorders, again, omics data analysis is actually proving to be very helpful, because traditional methods of observing histological data or biochemical assay data is limited in characterizing the tissues and samples in what they offer. And trying to extract the genes and trying to extract the gene expression data, trying to extract the DNA from within the cell is actually more helpful in trying to understand or trying to get a holistic picture of the samples themselves, which can be useful in relating to various different phenotypical applications and phenotypical characterizes of these samples that we have categorized. Like we can take samples from healthy patients, compare it to samples from who has developed chronic allergy to, for example, peanuts, and we can understand in what are the major ways the transcriptomics landscape differ in between these two samples, depending on where the samples are from. Samples could be from the skin, or from the stomach lining, or from the gut microbiome also. So various other omics data types are also explored in this biological approach. So all biology is not, all biology is not related or limited to being applied on Earth alone. So upcoming research in space omics, where NASA and other space agencies from all over the world are heavily focused and heavily investing, is the application of omics data analysis for understanding the effect of space on human body or on model systems. So people have found out effective method to simulate microgravity on model systems and microgravity on human cells themselves. So people observe, or people characterize these human cells based on the omics data analysis or molecular characterization that will take multiple different omics data characterization, and then compare the data landscape before and after subjecting the samples or subjecting the model systems to microgravity or to a space travel, in terms of astronauts, or in terms of even they took mouse to International Space Station once to perform these analysis. So these are the different fields that biology and bioinformatics and omics data, big data, omics data analysis are actually influencing actively and very rapidly, right? I mean, space omics is kind of a field that nobody would have heard about 3 years before, and right now it's actually picking up, and even a lot of upcoming and developing countries are actually focused on performing or understanding the effects of microgravity and space travel on astronauts and other subjects. So we have these different fields that are being actively explored on by students as they have presented you their research topic of interest. So it depends upon what you want to focus on. For example, you can focus on cystic fibrosis, you can focus on SARS-CoV-2 characterization using different analysis method. You can focus on long non-coding RNAs for being targets in leukemia, and you can also focus on different biomarkers, trying to understand biomarkers for prostate cancer or cancer types that is not being studied actively, like cervical cancers, prostate cancer, and etc. So this doesn't stop at the framework or at different biological aspects that we are limiting you to. You can also trying to mix match these analysis methods and different biological questions that you can answer from these. So I encourage you to look or explore this page, and this web page and web link has a lot of information and links forwarded for you to understand the effect of research fellowship program and its reach through the aspects or through the dimensions or through the view of students themselves. You are free to check different submissions from students and the variety of topics that they were interested in exploring omics data analysis and their applications in general. So with that, yeah, we also have furthermore links to some of the major or some of the key talks from compiled from our previous webinars and from a symposium that we successfully conducted at the end of or at the beginning of April rather.